Despite the development of antibiotics and vaccine approaches, infectious diseases still belong to the most important causes of human disease and death. This includes diseases that have plagued us for many centuries, but also newly emerging diseases or constantly evolving pathogens. Besides being medically important, pathogens also are fascinating objects of study. They've co-evolved with their hosts for many centuries, and we can learn a lot from them about the normal functioning of cells, tissues, and organs, and about the immune defense and the countermeasures pathogens have evolved against it. Generally, pathogen study is done in artificial culture systems to analyze their replication and spread. This includes the analysis of millions of viruses, bacteria, or parasites in a bulk culture system. This does not allow to study an individual pathogen or even the replication step of one single pathogen, however. Furthermore, the analysis is commonly done in artificial culture systems that do not completely reflect the natural infection of a host cell or host organism. Within Sonderforschungsbereich 1129, we want to take the investigation of important human pathogens to a new scientific level. Our central aim is the analysis of the interaction of pathogens with their host cells on a quantitative, single-particle, single-step analysis in increasingly complex systems to mimic the natural infection of the host organism. The key word here is the word integrative. We need new experimental approaches, we need new methods. These will be developed and validated in a close collaboration of infectious disease researchers with chemists, physicists, nanotechnologists, computer scientists, and mathematicians, but always directly associated with the infectious disease question analyzed. To foster this intense and direct collaboration between different disciplines, a new institute for integrative infectious disease research is currently being built on the campus with funding from the federal and state governments and the University Hospital Heidelberg. At the center of our approach is microscopy of the pathogens and labeling of the pathogens for microscopy to study their interaction with the host cell at the highest temporal and spatial resolution. Integration for us also means integration across many scales. We want to analyze individual components of the pathogen at the nanometer scale, but also infection of organs on the centimeter scale. And lastly, integration also means to link studies on artificial simplified systems with studies on individual cells, on complex mixtures of cells, up to natural or artificial organs. In the center of our scientific investigations are important human pathogens that cause AIDS, malaria, hepatitis, influenza, or dengue fever. All of these are important medical problems that need our attention. At the heart of Sonderforschungsbereich 1129 is basic research. We plan for the first time to follow an individual pathogen from the time of infection of a cell through all the stages of its replication and spread, but also its interaction with the immune system on a high temporal and spatial resolution and in most native conditions. Based on these experimental results, we also plan to mathematically model and simulate the individual steps of pathogen replication and spread, and we hope that this will allow us to form new strategies to fight against these important human diseases. In this approach, the Sonderforschungsbereich builds a new strategy in infectious disease research, and Heidelberg, with a strong interaction of the university and university hospital with its non-university partners, is the ideal place for such an endeavor.